and welcome to the next lesson for Advanced Higher Biology. This lesson is going to follow on really immediately from our last lesson, which was looking at the synthesis of the different types of proteins available in a cell. And this one really looks at what happens immediately after that synthesis. How are those proteins moved around the cell or secreted to the external cellular environment? So what we're looking for you to be able to understand from this one is be able to describe how proteins are transported out of the endoplasmic reticulum, explain how they move through the Golgi apparatus, state the processes that are going to occur as protein moves through, and name some types of secretory proteins and describe the movement of those proteins from them being translated to when they're secreted from the cell. So if you remember last time, we finished off looking at what happens when a protein, a polypeptide chain, is synthesized by the ribosome. And remember, we looked at the fact that there was a short signal sequence present on that polypeptide that was going to tell the ribosome where that resulting protein is to end up. And if that protein is going to form part of an intracellular membrane, or it's to be secreted from the cell, then it gets taken to the endoplasmic reticulum, where translation finishes off and the resulting polypeptide is released into the lumen of the endoplasmic reticulum. Any protein which completes translation in the endoplasmic reticulum is transported by vesicles, which bud off from the ER, so those little vesicles appear from here, and they fuse with the Golgi apparatus, and then they undergo post-translational modification before being secreted elsewhere. So within the Golgi apparatus itself, as proteins are moving through this Golgi apparatus, they're undergoing post-translational modifications, which primarily are the addition of our carbohydrate groups. If we look over here, this is our little vesicle that would be carrying the protein from the endoplasmic reticulum. It's arrived at the Golgi apparatus, and as the molecules are moving through the Golgi apparatus, they're moving from disc to disc. So remember, these are our menstruals that we spoke about before. If that's our, each one is stacked on top of each other with a little bit of fluid in between. And the vesicles are going from one to the other, going in very, very small little vesicles from this disc to this one, to this one, to this one, and so on, until all of their post-translational modifications are complete. And normally what's going to happen is enzymes are going to be catalyzing the addition of various sugars at different steps. So they might be adding a sugar here in this disc, another one in this one, and so on until the complete carbohydrate has been added to that protein and it's ready to be removed from the Golgi apparatus. So from that point, a new vesicle will form, and it will be taken, rather than going on to the next disc, it will be taken away from the Golgi apparatus, being shipped off and taken, in this diagram it's to the plasma membrane, but it also might be elsewhere, which we'll look at in a second. So the Golgi apparatus is often thought of as being like the warehouse of the cell. So it's receiving packages from elsewhere, repackaging them, repurposing them, adding any extra information it needs to those before it ships them off elsewhere so they can get to their final destination. So as I've said, this diagram sort of shows one which is being removed through the plasma membrane, but that's not always the case for these proteins. We also have some proteins which may be forming intracellular membranes, so they might be part of the membrane surrounding a mitochondria, for example. And if that's the case, similarly, we still have we've had our ribosome, we've had our endoplasmic reticulum. It's reached the Golgi apparatus, and what the vesicle does in this case is, rather than floating off to find a plasma membrane over here somewhere, what it actually does is it fuses with these long strands which are running through the cell, and these are referred to as microtubules. When that's the case, the vesicle moves along the microtubule almost like a train moving along a track until it reaches its appropriate destination. When it reaches the appropriate cell membrane or intracellular membrane that's going to be releasing the protein to. The vesicle fuses with that membrane and releases the protein. And as I say, that protein may then become part of the membrane of that particular cellular organelle. Just as a quick note from this diagram, you don't need to worry about what the centrosome is for the moment. That's not important for our course. It is there, it does exist as part of the cell, however, it's not something that you guys need to know about specifically. It just happens to be showing up on this diagram, which shows you quite nicely how the microtubules are passing through the Golgi complex, going through it and past it, so that those vesicles can come along and attach to it and move through the cell to get to the other membranes that they might need to reach. When we look at any proteins which are to be secreted, as we've already looked at, they are going to be finished translated on the rough endoplasmic reticulum. So the ribosome has acknowledged that signal sequence being formed on the polypeptide. It's taken it to the endoplasmic reticulum, forming rough endoplasmic reticulum, and we've completed the synthesis of that polypeptide, and it's been released into the lumen of the endoplasmic reticulum. So just the same as in our previous proteins. From there, it's passed through, the vesicle has transported that polypeptide onto the Golgi apparatus, and it's gone through its various different stages in the Golgi apparatus. Now that still might require some addition of some carbohydrate molecules, some post-translational modifications as it goes through. When it gets to the end, it is packaged into our vesicle, and in this case it's a transport vesicle that is going to take it to the plasma membrane. They're going to fuse with the plasma membrane, and it's going to release that protein out into the rest of the organism, depending on what type of cell this is. Now, this often happens with digestive enzymes or peptide hormones, for example, are good examples of proteins which might not be secreted. They might not be required for their cellular process. So in this case, they're going to be secreted and released from the plasma membrane. When this happens, often secreted proteins are synthesized as what we refer to as inactive precursors. So a really good example of this would be a digestive enzyme, which would be quite damaging to the cell if it was in its active state. Now, we previously looked at insulin as being an example of a protein which is inactive until it undergoes some kind of activation process, and usually that is proteolytic cleavage. So that was our other example of a post-translational modification, was the cleavage of that protein. So often what happens is the protein is produced as an inactive enzyme, secreted, still inactive, so it gets released through the plasma membrane, and it's not until it's required that it is cleaved and turned into its active form, where it then can carry out whatever processes it needs to, and usually quite quickly afterwards there's a natural inhibitor around which is able to prevent that enzyme from acting on anything else. So as I said, a digestive enzyme is a good example of that, because we don't necessarily want them active anywhere else in the cell. We want them to be only active and only acting on the types of molecule that we really need to break down, and not breaking down anything else that is vital to the survival of the organism. So from that, you should be able to say that proteins are transported from the endoplasmic reticulum and vesicles, that proteins move through the Golgi apparatus by moving from disc to disc through the transport vesicles, and the processes that occur as the protein moves through would be post-translational modifications, including the addition of sugar molecules or carbohydrate molecules to the protein. Types of a secretory protein would be a digestive enzyme or a peptide hormone, and they are going to move from translation to secretion by being translated on the rough endoplasmic reticulum and then secreted through the Golgi apparatus out through the plasma membrane of the cell. As ever, please feel free to ask any questions as we go through this later on, and make any extra additional notes as we go.